Hey guys, I wanted to kind of give you an update of what we've been doing here in the lab. So last time I showed you kind of that we did these vials, and then we also did these count plates. So what we found in our count plates was that the area outside wasn't suitable for drinking water, but then we took these and we then grew them on some other Petri plates, which these are Petri plates, and inside there you can see there's kind of a substance in there that's basically beef broth jello or chicken broth jello, and what that does is that allows bacterial colonies to grow, and so you can see on this plate, if I get behind it, it probably shows up better. All of these little dots are bacterial colonies. And even these little bitty ones down here. But basically all of those little dots, each one is a bacterial coliform, right? And so what we did, and this is another example, sometimes they can just look different on the plate. But these are, we basically took these and we put them on a slide, and then when we got them, we did a process called gram staining to them, which colors them so that we can see them in a microscope. And I easy basically by showing you a picture from my phone probably. So this is kind of what we saw when we looked at the bacteria in the microscope a little bit closer. So basically what we were looking at there then, we went in the lab to basically take what we had been finding in this stuff to try and see what it looked like, and if we can find its structure and then what it looks like, then that gives us kind of the first step of what we need to do to try and identify what type of bacteria it is, or what species of bacteria it is. And so by coloring them today, we were trying to answer 4 questions really, or a few questions. What color are they, pink or navy blue? And then what shape are they, circles or rod shaped or hot dog looking shaped? And then do they tend to want to look like they form chains or clumps, or we could even go neither. And then by having that information, we can figure out what the structure of the bacteria is. So if it is pink, it has a thin cell wall. If it is navy blue, it has a thick cell wall. If it is round, it is a cockle bacteria. If it is rod shaped, then it is a bacillus bacteria. And then if it likes to form chains, then it is strepto. If it clumps up together, then that is staphylo. So what we found in that picture that I just showed you is there were some examples that were hot dog shaped bacteria that were pink, and they like to form chains, and usually they only chain up in pairs. And so what that suggests, but does not necessarily prove, it gets us to a point where we have pink. This whole staining process was invented by this guy named, I think it's Christian Graham in the late 1800s. Anyway, if it's pink, it has a thin cell wall, which is basically, we're calling it negative for a thick cell wall, so it's called gram negative. And then if it's hot dog shaped, that's bacillus. If it's chains, that's strepto, so it is gram negative streptobacillus, right? So gram negative streptobacillus. Now that is just a morphology of a bunch of different bacteria, but E. coli happens to fall under that morphology. So if we actually arm ourselves with this, whatever happened here, and then finding pink hot dogs, that kind of tends to paint a broad picture that we are probably dealing with E. coli. but there's 3 or more tests that we would need to do to get to that point. Now, do you need to know this for a quiz? No, not necessarily. I just wanted to show you what we're doing here in the lab because it relates back to Snowmageddon that we were talking about before. We do this and this kind of at the same time, and those take 24 to 48 hours to get results. If we were to come back with a result like this, we would obviously say stop drinking the water or keep boiling the water, right? But we would also want to identify what pathogens or coliforms were particularly in that sample. And so to get to that point, we would have to go about another 48 hours. And then that would get us to the point where we got today, where we said, okay, we have gram-negative Streptococcus, which those bacterial morphologies then kind of narrow it down into where we can do a bunch of different tests to figure out exactly what species of bacteria we're dealing with. So it's kind of like it gets us to, kingdom, phylum, class, order, family, genus, species. It's kind of like it gets us to phylum, and then we've got to go another test to get us to class, and then order, and then genus, or whatever. So that's kind of, we did that here to show that at best, we're 3 days in and we've just identified how we're going to test more to figure out what it is, right? And so it's a lengthy process, and then these tests that you have to do are going to take, several more days. So it could be, up to 2 weeks before you even have a result, possibly more. So really it's showing you that these tests take a long time. And then not only that, but this stuff doesn't grow on trees, right? It's expensive. And then we don't just test like, a few places around the school. If this is a city, we're talking about someone, needs to go all the way over to the north end of town, the south end of town, the east end of town, the west end of town. So you got to go get samples from several different locations. So you're talking about man hours and manpower to go get those samples. So really this isn't feasible on an everyday basis. Usually it is kind of a reactionary measure to what has happened. So someone gets sick, well, let's test the water. Or the pressure's lost in the pipes, so let's test the water. Is it a broken line somewhere? So that's kind of why I wanted you to show you this. And then before I forgot to tell you that, so this applies to all bacteria. So you've probably heard of, I have strep throat, right? Well, the bacteria that causes that infection in your throat is a strepto. I can't remember if it's cocci or bacillus, but a streptococcal infection in your throat. So when you go to the doctor, they swab your throat, they put it on a petri dish, and their petri dish is designed to actually only grow that particular bacteria that causes you the strep throat. So usually, if after 24 hours or however long there's no colonies growing on this, then you don't have strep throat. But if there is colonies growing on this, then you probably have strep throat. I think they have more rapid ways to test for it today. But that's basically what they do. They test to see if the streptococcal bacteria are present in your throat. If they are, then you've got strep throat. So why do they call it strep throat? It's a streptobacteria that is infecting your throat. You've probably heard of staph on your skin. If you get a staph infection, that's a flesh-eating bacteria that eats away at your skin. I don't know if it's Staphylococcus or Bacillus, but it's one of those. So that's kind of what those names lend themselves to, and how it kind of applies to the real world. So what are you going to be tested on? None of this, except that fecal coliforms can be tested for. It's expensive. It is time-consuming. It requires a lot of effort. So it's not always feasible. It's a reactionary measure, usually to something that we found to be a problem. That's where we've been in class doing some lab work. So Unit 9 should wrap up a little bit quicker now. So be ready for that tomorrow and going forward through the rest of this week. That's all I have for now. So I will talk to you soon.